This Week in Microbiology is brought to you by the American Society for Microbiology at microbeworld.org/TWiM. This Week in Microbiology, episode number 1, recorded February 18th, 2011. Hi everybody, I'm Vincent Racaniello, and this is the first episode of TWiM, the podcast that explores unseen life on Earth. Joining me today to talk all about microbes is Cliff Mintz. Hello, Cliff.
Hello, Vincent.
Welcome to TWiM.
Thank you very much. It's a pleasure to be here.
Glad you could join us. You are currently a freelance writer, blogger, and speaker. Is that correct?
That is correct. I'm a recovering scientist.
Well, we'll have to hear about that. Also with me today from sunny South Carolina is Michael Schmidt.
Hello, Vincent. How is everyone this afternoon?
Not as warm as you are, as I understand.
Yeah, we have what they say in the business is Chamber of Commerce weather today. So I'm very happy to be inside reporting on the unseen world of microbes.
Glad you could join us. You were at Medical University of South Carolina, is that right?
Yes, that's located in Charleston, South Carolina.
Very good. And our third co-host today from Washington, D.C. at the moment, but normally in San Diego, is Stan Maloy. Welcome, Stan.
Hi, Vincent. It's wonderful to be here.
You're at San Diego State University, right?
That's correct.
All right. Well, this is the first episode of TWiM. It's a historic podcast. And we're going to tell you about what we're going to do on TWiM. The idea is to talk about microbiology. And according to Wikipedia, microbiology is the study of microscopic, unicellular, and cell cluster organisms. And they say this includes eukaryotes such as fungi, protists, and prokaryotes. But they did forget the archaea, and I think we'll certainly talk about those as well. And my idea is that we'll talk about all sorts of aspects of microbiology, and we'll talk about microbes that make you sick, we'll talk about microbes that help you, the ecology of microbes, how we use them, how we prevent infections. And I suppose from time to time we should also talk about viruses, especially those that infect bacteria. So that'll be the general idea. We'll pick stories every week, and I'll tell you at the end of the show how you can get involved in selecting those stories that we discuss. And before we start today, I would like everyone to tell us a little bit about how they have arrived at where they are today. What's your background, your educational background, your research background, and so forth. And let's start with you, Cliff, since you're a recovering scientist.
I have a PhD in bacteriology from the University of Wisconsin-Madison. I did 5 years of postdoctoral studies. I've studied Salmonella typhimurium, Neisseria gonorrhoeae, and Legionella pneumophila throughout my graduate and postdoctoral training. I was a professor at the University of Miami School of Medicine from 1987 to 1994. I left academia in 1994 and have done a lot of different things for the past, 17 years. And currently, in my most recent incarnation, I'm a freelance science writer, medical writer, journalist, and social media enthusiast.
So are you recovered yet, or you're not yet recovered?
I've sufficiently recovered, but I still enjoy a little tete-a-tete surrounding science and its relationship to reality.
So I think you can give us also the industry perspective, because you were in industry for a while.
Yeah, I've worked in industry as a consultant as well as an employee, and so I do have that perspective.
So we have to do a little disclosure here. Cliff and I both went to Cornell as undergraduates. All right, so you're an old friend of mine, and in fact, you were here as a postdoc for a while with Howard Schuman.
Yes, that's exactly right. When you were a assistant professor, I was still a postdoc, but let's not talk about it.
Okay. So from that, we can tell why Cliff is a good member of the TWiM team. Let's move over to Michael Schmidt. How did you get to South Carolina? What was your path?
My path, I started at the University of Illinois in Chicago, where I got a degree in biological sciences. And this is one of the instances where when you have a great mentor, you can actually pick a career path that interests you for the rest of your life. And my triggering point was a TA that I had for my first microbiology class. And she encouraged me to think about microbiology as a career. I was at the time working on an undergrad research project on cave spiders. And from cave spiders, I evolved into microbiology, where I went to graduate school at Indiana University in Bloomington, where I obtained a master's and PhD in microbiology under Walt Konietzka. And Walt had a really great philosophy about training graduate students. You'd put them into a room and you'd give them everything that they could eventually want to need, and eventually they'd find the door and graduate. And it was really great training. I made a lot of mistakes, but I had a lot of successes. And from Indiana, I left Indiana and went to the State University of New York at Stony Brook, where I did a postdoctoral fellowship in the laboratory of Don Oliver working on bacterial protein export. And from there, I went down to South Carolina, where I became an assistant professor at the medical university, where I've had a very eclectic research portfolio for a faculty member at a medical school, where I worked on everything from bacterial protein export in E. coli, Listeria, tuberculosis, to I developed in conjunction with a chemical engineer a membrane bioreactor that could degrade solvents in real time, which was a pretty neat learning experience. It's again where you have an opportunity to be mentored by a senior faculty member. In this case, it was a chemical engineer. So I've been doing translational research probably for the 20 years that I have been at the medical university. And I evolved from membrane bioreactors. I did a stint working on HIV for where a colleague left the university, and I inherited graduate students. And my current research interests right now are hospital-acquired infections, which is a substantial issue for the country. So as I tell anyone who asks me, each day as a microbiologist is an adventure, and it's a really rewarding career path because you really stimulate it each day.
Good path. I know a faculty member out at Stony Brook, you probably do also, Eckhard Wimmer.
Eckhart is a treat. I really enjoyed my interactions with him when I was at Stony Brook, and you know, he's one of the great polio virologists.
That's right. We have butted heads on a number of occasions, as you might imagine.
Vincent?
Yes, sir.
Could I point something out here?
You may.
It seems to me that there's a strong John Beckwith thing going on here.
What's that?
Because I believe that Howard trained with John Beckwith, and I believe Don Oliver did, and I don't know where Stan did it, but Stan somehow fits into this also, I think. I may be wrong.
Well, actually, the great story about Don Oliver is he actually got his PhD from Elio Schechter and then went and did his postdoc with John.
Because Don was in Howard's lab at the time I was a postdoc doing research when he's still at Wesley. He was at Wesleyan, I believe, or something. I don't remember.
Yeah, Don's still at Wesleyan.
Yeah, so it's a small world, so to speak, in microbiology.
Stan, what were you going to say?
I was going to say I never really trained with Beckwith, but once upon a time I took the advanced bacterial genetics course from Tom Selhavi, who is a Beckwithite.
Right,
and that
right.
changed my life.
Okay.
Yeah, so this is typical of science. It is a small world, and you can get 10 people in a room, and besides 3 of them having the same birthday, a lot of them will have worked for the same people. Stan, what about you? What's your path?
Well, like Michael, I started off with the goal of becoming something else. I was all set to be a bioanalyst and took a microbiology class when I was a senior in college. And I said, damn, I have to do this. So I quickly switched gears. I went off. I got a master's degree working on an unusual freshwater bacterium called Microcyclus. And from there, went on to work on E. coli lipid metabolism and central metabolism. And I graduated with a PhD in biochemistry from UC Irvine. I did a postdoc in bacterial genetics with John Roth at the University of Utah. And then I was on the faculty at the University of Illinois in Champaign-Urbana for almost 2 decades and moved to San Diego in 2002. And I've been at San Diego State since then. My research is focused on chromosome rearrangements in Salmonella and evolution of host specificity, particular questions like, why do some salmonella only infect one kind of animal and other salmonella can infect a broad range of animals? And because of students dragging me there, we've been doing a little bit of metagenomics as well. I've been really lucky because in the process of all of this, I was the president of the American Society for Microbiology, which was a great experience. And in addition to academia, I've also dabbled in biotech, ag tech, and pharma. So it really has allowed me to interact with a lot of people and see the wealth of things that microbiology can impact.
Well, that's something we hope to explore on this podcast. And as you can see, everybody's well suited to do that in different ways. It's a great crew. And in fact, we expect to have other people join us in future episodes as well that'll complement this. I am here because I love using social media like podcasting to teach about microbiology. I have 2 other podcasts, This Week in Virology and This Week in Parasitism. And I am a virologist. I have been working at Columbia University on virology for about 30 years now. And so my contribution to TWiM will be to ask, as Dick Despommier says, to ask the dumb questions, because I don't work on bacteria, except to clone genes in them, and that's minimal. So that's today's panel. And Stanley, before we go into a story, I think you're at the AAAS meeting. Maybe you could tell us if there's anything interesting going on there.
Oh, absolutely. Although if I could just back up, I think us microbiologists consider virologists to be a subset of microbiologists. So you're a bona fide microbiologist.
You think so? I wanted to ask you that because some people think that virology is not part of microbiology, which of course is absurd because I'm in a microbiology department here.
Right.
But we will certainly talk about viruses. We're going to have our listeners suggest what we talk about, and if they tell us to talk about a virus, for sure. Over on TWiV, when we're asked about bacterial viruses, we often are not very knowledgeable because none of us work on them. So maybe that would be something we could focus on here.
That'd be great. Well, so you asked me to talk about the AAAS meeting, and this is a very broad and big scientific meeting. But the thing that's really striking is that we started off the first day of the meeting today, and one of the first sessions was a session on microbiology, and it was the most packed meeting in this whole place. That meeting was focused on the microbiome, and the microbiome is something that there could be a lot of discussion about because it's just one of these mind-blowing things where somebody develops a new technology and all of a sudden you see impact in many different ways.
So by the microbiome, you mean all the bacteria, archaea everywhere?
Yes, that could be all of the microbes everywhere. In particular, I think in this group, they were talking about those that are associated with humans. So those that live on our skin or in our mouth or in our gut.
Okay, because I know there are efforts to understand the environmental microbiome as well, right?
Yeah, absolutely. And in fact, some of the microbes move back and forth between the two. So
do we need to further know the microbiome in us, or I thought we had characterized it, or only in a limited way?
Well, so this is one of the reasons why it's so important at a meeting like this. There's been so much discussion since we had human genome sequencing that we would have personalized medicine, and that if you could see my genome sequence, you could make predictions on how I should be treated. And the recent work has suggested that the microbiome, the microbes in us, particularly in our gut, have a huge impact on our health. And so personalized medicine would really need to integrate the microbes, not just the human genomes.
So each of us has a unique microbiome.
Absolutely. So for example, one of the talks this morning was a talk by Jeremy Nicholson. He's a professor at Imperial College in London, and he does metabolomics. That is, he looks at the types of metabolites, the biochemistry, focused on humans primarily. And in their studies, they discovered that the microbes that are in the gut play a key role in modifying some of the metabolites that are made by humans and are critical for human metabolism. So it really is a symbiosis where humans have the enzymes that allow them to make some of the products they need, and the microbes modify those products and make a whole bunch of other products. So
what's amazing to me is that we didn't appreciate this. How many years ago?
right.
5 years ago, maybe. We had no knowledge, and the ability to sequence has given this to us. So I'm sure we will be talking about the microbiome in many future TWiMs. Is there anything else at AAAS of note?
Well, so one other thing that we talked about this morning that I think is really exciting is a series of efforts to be able to predict where the next outbreak of an emerging infection will come from. And the key thing about that is that it's not a bunch of microbiologists or a bunch of epidemiologists who are doing it. It's really integrating new media, cell phone technology, Twitter, and a lot of satellite imaging. So it's a great example of the integration of all of these fields to be able to achieve a scientific goal.
Sounds like you're on top of new media, Stanley.
We have to all be nowadays.
We have to, but not everyone is, unfortunately. I know you have to leave in a few minutes, but I thought we could start talking about one of our two stories. Maybe you could chime in early on. The one paper that I picked out this week that I thought was interesting, it's in MBio, which is the open source ASM journal. It's called Opportunity and Means, Horizontal Gene Transfer from the Human Host to a Bacterial Pathogen. And it's by Mark Anderson and Steve Seifert. And it basically describes the presence of human DNA integrated into the genome of Neisseria gonorrhoeae. I don't know if any of you have taken a look at this. I know, Stanley, you have communicated this paper to the journal, right?
Yes, that's right.
Any thoughts about it?
Well, I think this is one of those examples that we were just mentioning another one earlier, that it's such a surprise because we really didn't have the tools to see this stuff going on earlier. But the basic idea is that this is a chunk of human DNA that's plugged into Neisseria gonorrhoeae, and it's absent from other closely related Neisseria strains. So the suggestion is that this has happened in very recent time.
And this transfer of human DNA to Neisseria would have happened in some human host because the sequences they examined were clinical specimens.
Absolutely. So this is a case where the pathogenic bacteria are snuggled up close to their host, and the opportunity for taking up some host DNA is pretty high, but to integrate it into the Neisseria genome and then maintain it implies that it might be doing something. And that's where we still have a huge question. We don't know if it's still functioning, or if so, what the heck it's doing.
So are there other examples of human DNA sequences in bacterial genomes, or is this the first?
This is the first example that I'm aware of.
Okay. So this could be very special.
And if I'd like to interject, I think this is going to go down as one of those seminal papers and really spur the people who are looking at pathogenesis for those organisms in which we're seeing higher and higher carriage in human populations, like MRSA or methicillin-resistant Staph aureus. That microbe has really chronically infected hospitals and their patients for a long period of time, and now it seems to have moved out into the community. So when I read this paper, the first question I had is what to hunt for in the staphylococcal genome that could suggest this is how staphylococci, specifically MRSA, may be interacting in the nares of humans to effectively establish itself as a good colonizer, because that's really one of the great mysteries of pathogenesis, is the whole issue of how these microbes establish themselves on people. And I think this paper especially will give us those methodologies to begin to go on this expedition to find how and whether or not horizontal gene transfer is just isolated to Neisseria, or whether it's isolated to those other pathogens that are causing a substantial amount of disease in humans.
This is something that everyone can do because initially it's just a matter of scanning the sequence databases for human sequences, which is what they did. There are 14 different clinical isolates of N. gonorrhoeae sequences, and they just looked in them for human sequences, and they found human sequence. And the particular piece of gene, of DNA, it's a 685 base pair fragment. It derives from what is called a line, L-I-N-E element. It's a human retrotransposon, and line stands for a long interspersed nucleotide element. Is that right? Let me see if I got that right. I know it's long interspersed. I don't know what the N stands for, if it's nucleotide or...
It must be nucleotide.
Nuclear element.
Nuclear
element.
Long interspersed nuclear. Here I am. I'm looking at my textbook where I write about it, and I don't even know it. So lines are derived from retroviruses. They are retroelements. They have bits of retrovirus sequences in them. So this is appropriate that I get to talk about a virus on the first TWiM here.
Well, the thing is also, Vincent, it integrates into a Neisseria gonorrhoeae prophage sequence. So, we're right in the heart of the virology story right now.
Yeah, and I
can't get away from virology.
Well, I don't want to, but of course, I picked this story, so in the future we won't always be doing this. But it is a retrovirus-derived sequence.
The other interesting thing about this, Vincent, is if you go and ask the question about pathogenicity islands, which are ubiquitous amongst the pathogens, the operational definition of a pathogenicity island is it's present in pathogens and absent in non-pathogens.
Right.
And anytime you see these repeats, it effectively serves as a landing site for genetic information that may confer a selective advantage to the bacterium. And the question is does this line element really confer a selective advantage to these Neisseria in adapting to the human host?
Right. I think they note, that's a very good question, and they note that they compared strains with and without the element, and at least in the lab they don't see any difference. But the real key is in people, of course, whether it makes any difference to have this.
Well, the interesting thing about this is that if you look at what they did, they only found that 11% of the clinical isolates had the element. Mm-hmm. So the question that I would have as a recovering or former pathogenesis guy is, was there any relationship between virulence or types of disease caused by the clinical isolates as compared with one another? For example, there are certain strains of Neisseria that can lead to pelvic inflammatory disease. They get into the bloodstream a lot more efficiently and effectively than the ones that just cause gonorrhea, which is relatively a localized infection in the cervix. So, 11% is relatively low, but without understanding, what types of diseases, where they came from, it kind of, maybe that's the next step.
In many ways, this is a complicated problem. It's kind of like the drunk looking for his keys under the light post, even though he lost the keys somewhere else. There are so many different possible places it could have a role in fitness, right. And the assays we have are so limited that it becomes very difficult a priori to make that judgment call.
Right, and the important thing to point out for people out there when we talk about animal models of disease, there are no good animal models of disease for Neisseria gonorrhoeae that are good to study the true pathogenesis. So we are really limited to the types of studies that could be done in vitro with animal models that really aren't representative of diseases that are caused in humans.
So what you have to do is epidemiology to see if the presence of this human sequence tracks with any significant disease difference.
Right, and I would submit that if these strains were isolated from women with women with pelvic inflammatory disease, that there may be an advantage there if they're across the board. Then maybe virulence itself, in an obvious way, may not be the trait that is affected. So,
right.
I don't know.
It's probably worth pointing out that this line 1 element, L1, comprises 20% of the human genome. right? 20%. There are something like 10 to 100,000 copies of this line element in human DNA. So it's very abundant, and that might be why, in part, it's in this bacterial genome.
So another aspect about this particular bacterium is that this bacterium is very naturally transformable. That is, it's really good at sucking up DNA from the environment and bringing it into itself. So, if you were to guess where you would think this might happen, Neisseria is one of those places.
Yeah, and that was a point that I was going to make too, is that it's one of the best, if not the best, natural transforming organism. That is the ability of bacteria to pick up DNA out of the environment and incorporate it into its genome, and it is that. And given what goes on in a localized gonococcal infection, there's a lot of cell destruction. In the paper, they speculate there could be an apoptotic event, and there's lots of host DNA out there, possibly bathing organisms, gonococci, in or fragments of DNA that are transformable. And if there's a frequency and maybe if there was some kind of restriction, modification, mutational event in a certain strain, it may have found its way into that clone, and then ultimately the strain did affect the fitness of the strain, and it was just incorporated for whatever reason.
The puzzling thing that I had is why it doesn't occur in the other Neisseria, like meningitis, and the other normal flora that's part of the upper respiratory tract. I found that pretty intriguing. Is it because the meninge doesn't really cause a fulminant infection per se in an individual, and it's a chronic colonizer, so it has no need to pick up the DNA? Or is there some fundamental issue about how L1 facilitates fitness of gonorrhea over the meninge or the other Neisseria that are normal flora of humans?
Yeah, well, it may be that we haven't looked enough. Maybe as more isolates are looked at, it'll be found, right? Because we only have 14. So,
there's one other thing about this, and that is, whenever people find a piece of DNA that they don't expect to find and it seems to be conserved, they say, aha, it must be important, otherwise it would have been lost. Well, maybe it's not so important, and it's just there by chance, and it just hasn't had enough time to be lost yet.
We always talk about that. Why is 20% of the human genome this line element? Why isn't it gone? We think it's useless, but who knows? It's the same issue. There is some evidence in this paper that this sequence is actually expressed as RNA. They have an experiment where they've extracted RNA and amplified it by PCR. It's amplifiable, so there seems to be some transcript made, which again could be accidental or it could mean something.
Well, it does. I guess it does insert within the prophage or close to the prophage DNA sequences. So if it's there, it's likely that there may be some upstream promoters or some elements that are.
Yeah.
It makes perfect sense to me that it would be transcribed.
Yeah. What's really would be important would be to know whether a protein is made. And then we don't know that from this work, but presumably they're working on it, making some antibodies.
Since we're all Beckwith disciples, we can go back and put a reporter cassette in and answer the question.
That's right.
Exactly.
So this is interesting, and we'll see what comes out next. What do you think is the next thing that has to be done?
Probably a scan of public health databases where they have archived Neisseria gonorrhea strains, and do a quick and dirty PCR to ask the question, what proportion of the archived strains do indeed have this L1 element? And ask the question almost retrospectively with an epidemiological survey, asking how many of these Neisseria, what types of diseases did they cause? Because the public health databases will report whether or not that patient hopefully developed either PID or whether or not it was just a normal run-of-the-mill STD that was easily cured with a course of ceftriaxone.
This is really funny because I have a very different answer, and it just reflects the fact that we do different kinds of science. And it's interesting how different scientists will answer that question in a unique way. What I would do first thing is I would go in and make a mutation in that sequence.
That's what I was going to say too, to see if it has any effect.
I would want to have those isogenic strains, the strains that are exactly the same except for that mutation, so that I could explore that at an early stage.
Well, what would you do with those two strains? If there's no animal model, you would be restricted to growth in the laboratory, right?
Well, so you could look at a variety of conditions of growth in the laboratory. That is a possibility, but there aren't good animal models, but there are.
There are, right, some.
Okay.
And so I think you could ask the question about fitness. And so the kind of question I would want to ask in that case is to take the two strains that were isogenic except for this mutation and some other marker, and to ask if you add them both at the same time to the media or whatever assay you're using, does one do slightly better than the other, implying a fitness advantage?
This also emphasizes why the microbiome is important, Stanley, because now when you get those sequences, you can look for human sequences in them.
Absolutely.
So this becomes a bioinformatic problem, and in fact a big one, because we're going to have a lot of sequences generated in the next few years.
But, just to raise a general point, going back to the whole mitochondrial eukaryotic cell thing, right? I mean, there is
Can you explain that to us?
Well, I mean, I don't know if this is true anymore, but this is what I learned as a microbiologist, that the fact that mitochondria may have in fact been ancient, bacteria that, somehow became part of a eukaryotic cell.
Right.
So to me, finding human sequences, particularly in a pathogen, like Neisseria, which is, an obligate human pathogen, the fact that evolutionarily there may be hints that it's not that surprising, and like Stanley and Michael, the only reason we didn't know about it was that years ago we couldn't do the research because we didn't have the tools.
Sure.
So I would not be surprised if you look at perhaps even intracellular organisms like Legionella or Rickettsia or some of the others, even though they're less complex from a DNA standpoint, you may be surprised. They live inside of cells, so, why not?
Sure. Well, it is certainly interesting. I look forward to the next part of this story, and probably will go on in different labs in different ways, as we've discussed.
Could I make one last comment about that paper?
Of course.
This is an interesting paper because, as you can tell from hearing us talk, there's more questions than there are answers. But for me, at least, that's what I find really exciting in science. When there's something new and it just stimulates you to think in different ways than you'd thought before and about different things than you'd ever thought about before. So
absolutely.
So I really like it.
Yeah, if it were all answered in one shot, it would be less interesting. But you're right. That's a good one. Well, thank you for contributing that, or being the editor, Stanley.
Thanks a lot. And I think we're going to sign off now. It was really nice talking to all of you, and I hope we have the chance to talk again.
Yeah, thank you, Stanley. Hope you can come back on another TWiM.
Take care.
Bye. All right, let us move on to the next paper, which is called Dry Copper Kills Bacteria on Contact. And I saw this, and I thought, Michael, you would like this because I know you're a copper guy, right?
Yeah, this is a really remarkable paper, and it really goes to an old observation. Mm-hmm. The observation that copper is antimicrobial has been known since the time of the ancient Phoenicians. There's actually stone tablets in the British Museum that actually describe the antimicrobial properties of copper, in which they tell you that if you're wounded in battle, what you should do is sharpen your bronze sword before you go out and battle the next day, and allow the shavings to reside in any wounds you might have.
Wow.
And that will effectively prevent you from losing your limb to a fulminant gangrenous infection. So the ancient Phoenicians knew of this observation. The ancient Egyptians used copper vessels to purify water or make it potable, and the recipe was quite simple. You put water from the Nile in a copper urn, and you would allow it to sit out all day, and then it would be safe to drink with your evening meal. And so this is an old observation, and metallic copper is the key. This is not truly copper in solution. And these authors did a heroic effort in demonstrating how it's dry metallic copper. And I think what was most fascinating, and this will help the casual listener think about how small bacteria are when they talk about the number of copper atoms that are actually inside the cell. To give the general listener an idea, the volume of a bacterial cell is 10 to the minus 15 liters. Everybody, if you ask casually on the street, what's the pH of a cell? And most everyone in biology or biochemistry will tell you, well, the pH of a cell is really about 7, give or take. And any freshman chem student knows that pH is a logarithmic scale, and a pH of 7 means there's 10 to the minus 7 moles per liter of free proton available, H+. So this teaches us that in the average bacterial cell, there are only 60 free protons. It even gets squirrelier when you do additional math, when you ask the question, what do flagellar motors spin at? And, they spin at they measure it, it's 100 hertz, which is 6,000 revolutions per minute. And in order to move a flagellar rotor, one revolution takes 1,200 protonic fluxes across the membrane. And that introduction gives you how important ionic flux is to a bacterium in terms of maintaining its charge balance, because we all learn the equation delta mu H plus, the famous Mitchell equation for energy. Mu H plus is equal to the change in delta psi, the charge, as well as the change in pH, or delta pH, or the chemical ion concentration. And what this paper really shows is that what the copper ions are effectively doing from the dry surface is are really mucking up the membrane. And they have some really neat techniques that they're able to show how it works.
The copper ions actually get into the bacteria. So if you put the bacteria on elemental copper, just metal surfaces, the bacteria take up the copper, right? Somehow it gets in.
I mean, the paper is speculating, but it's clear that it's a very immediate event, and the bugs are just, or the bacteria are just obliterated in seconds.
Which is why I went into the diatribe about delta mu H plus and this whole issue. I think, given that all living cells must pump ions for a living, and they have so few free protons in order to do this with, because bacteria make their living by pumping protons and electrons. They go out in pairs, if you will. And so the minute it sees, I think, the metallic copper, that electron, because metallic copper has a partial plus state, as they point out, the minute that electron sees it, the electron falls into the copper river, the metal river, and it's the equivalent of you trying to jump out of a swimming pool. You can do it, but it will require a tremendous amount of energy. And I think the instantaneous mechanism is likely due to the loss of the ionic balance in the cell, because anytime you collapse either delta psi or delta pH, you kill a cell. I mean, that's the classic Mitchell mechanism for generating energy as well as ionic transport, which is an absolute requirement for all living cells, ours, humans, as well as microbes.
So the disintegration of the cells that they observe on the copper is a consequence of that imbalance that you're talking about.
I believe so, and
because they don't really figure it out.
Well, how do you really tease it apart? Because those experiments are extremely challenging to do, because, again, 100 hertz just to spin a flagellum takes 1,200 protons to move one rotation when you're in reality at 100 hertz, you're spinning at 6,000 RPM.
Right.
So and I think that's why it's instantaneous and why they see such a dramatic amount of collateral damage to the cell. Mm-hmm. The other really cool thing they did is they broke out Deinococcus radiodurans, the most resistant microbe on the face of the earth. This sucker was isolated from waste tanks of our nuclear legacy, in which this microbe is constantly undergoing repairing of its DNA. And they were really able to discount that copper was actually damaging DNA in the sense that that was responsible for its mechanism of death, which had been proposed in the literature previously. Mm-hmm. So it's a very provocative paper that I think goes to try to explain the ancient Phoenician observation that metallic copper is good for controlling microbial growth.
The bacteria they use here, besides Deinococcus, is E. coli, but I presume the antimicrobial effect of copper is broad.
The antimicrobial property of copper is almost universal. There are a number of papers that have been published in this same journal, Applied in Environmental Microbiology, by the laboratory of Bill Keevil of the University of Southampton, in which he's been able to show it for a large number of microbes, including Clostridium difficile, that's in near epidemic proportions across the country. And Dr. Keevil's also shown that metallic copper can kill influenza virus. And that's a really neat paper if you haven't seen that one, Vincent. Metallic copper killing a virus, and I always have problems with killing viruses because they're not alive. So I like to use the word inactivate.
Right.
Unless you consider viruses alive. But I think it's really pretty neat on how the method they have gone about using some of these remarkable dyes to measure activities. So in that respect, it's a really good techniques paper, though I don't necessarily think they really get to the precise mechanism of how copper is killing. I think it's probably more multifactorial than their experiments can lead us to conclude.
So back in September, I think it was, you and I had a conversation about using copper surfaces to prevent infections. So is this being explored to a greater extent now, like you have?
Yeah, I think there are a number of laboratories out there that are trying to investigate this. I'm in the process of collecting data from a full-scale clinical trial at 3 hospitals across the country, in which we're asking a simple question: can metallic copper lower the incidence of hospital-acquired infections? And we are collecting data, and we're in the midst of the clinical trial. So we're in what is termed in the industry the silent phase, where we can't neither confirm or deny.
So these aren't copper's not at the moment being used in hospitals. It's just being examined for whether it is effective in that setting.
And I think what that articulates is the chicken and the egg problem of most translational research. Before the medical community is going to adopt a technology or a practice, they want to see the evidence that it actually does indeed lower infections. And, hospital-acquired infections are a substantial problem to the United States. One of the interesting things is HAIs kill more people than HIV and breast cancer combined each year. Yet there's no telethon, there's no marathon, there's nobody at the Academy Awards wearing a ribbon. The issue is that the individuals who die, unfortunately, die one at a time with their loved ones around their bed. And the patient often gets the blame, or the bacteria get the blame, because we either don't have a drug that can treat the microbe, or the patient is so ill that the microbe just overwhelms their system. And again, the poor patient passes.
I'm trying to visualize how you could use copper in this way. Like, I know you could put it on surfaces, right? But what else? Is there any other way that you could utilize it to be an antimicrobial?
Surfaces is probably your best bet, because we know the environmental contribution. We've all heard of the 5-second rule with chocolate chip cookies falling on the floor.
Sure.
In a hospital, there's a study that has been out that when clinicians touch a bed rail that has been contaminated with VRE, vancomycin-resistant enterococci, you can actually transfer the VRE from the surface, which most major hospitals are using plastic bed rail, from that plastic to the healthcare worker within less than 5 seconds. So the 5-second rule does not apply when touching objects in the built environment of a hospital.
So could I interject something and just ask a question? So back in the old days when I took introductory microbiology, there was this assertion that most metals are antibacterial, that you don't necessarily have to worry about bacteria persisting long on doorknobs or other types of metals. So they did use stainless steel as a control in this paper and saw that stainless steel, unlike copper, didn't have that effect of instantaneously killing and destroying the metallic copper. Metallic stainless steel didn't kill the bacteria, either the E. coli or the radio resistant strain, right? So wouldn't it have been more informative if they may have used another metal as a control rather than steel, or is they use steel because steel is iron and some other metal?
I think the reason they use stainless steel is the literature out there shows that if you put 10 to the 8 bacteria on a stainless steel surface at the end of the observation period, which is typically measured in days, you at most have lost 1 log unit of the bacteria. While if you put it on an antimicrobial metal like copper, within 2 hours you kill 99.9% of the microbes, and in some cases you can actually kill 7 logs of methicillin-resistant Staph aureus within 90 minutes. So I think they were trying to demonstrate the issue of in that particular control that they use, that it wasn't, I guess, mutagenic. I think that's what they were trying to say in that particular figure.
I see.
Well, yeah, and the other metals that are out there, you have silver, you can, use zinc, right. and there's a whole series of alloys that you can get around. And I think most of the alloys that we learned about when we were young graduate students always had enough copper in them to effectively provide the antimicrobial activity.
So it's interesting that you mentioned zinc. Does zinc conclusively have antibacterial properties in any kind of therapeutic environment that you know of?
That I don't know of. I have not seen any data out there on zinc in terms of its antimicrobial kill. I know zinc can be alloyed with a number of these metals, but I don't know if anyone's actually used zinc per se, and I don't know whether or not zinc is specifically an engineering metal or whether or not it has to be used in concert with copper and other things. Because as you know, our pennies are zinc.
Yes.
Is that right? There's no copper in a penny any longer?
No, there's copper in a penny, but zinc is less expensive than copper, so pennies being only a penny, they have zinc in.
So that actually brings up a very interesting point. So you're talking, Michael, about using copper as a antibacterial or antimicrobial in a hospital setting. From a cost standpoint, if copper is that expensive, and now it's bringing back the notion that people were going out and stripping houses of copper wire to make money because the cost of copper has gotten so high. From an antibacterial standpoint, it would make a lot of sense, even if copper showed a marginally positive effect on reducing the numbers of hospital-acquired infections. But the question is, would hospitals be willing to spend the money to retrofit their hospitals with copper, given the expense as compared with plastic? So in the laboratory, it makes sense, but in reality, would copper really be adopted?
The answer to that question is probably yes. There are a number of reasons why. The average length of stay in a hospital is approximately 5 days. If you develop an infection and you have a 1 in 20 chance of developing an infection, you increase your length of stay to 24 days. That adds on average, using most current data, $43,000 to your hospital bill. It costs the United States individually $47 billion in added cost. So if you could move the infection rate a decimal point, you could save literally billions of dollars. The other reason that I think hospitals will beat a path to any material that will cut the infection rate is the Center for Medicare Services, as well as HHS and the new healthcare law, have specifically incentivized hospitals to lower their infection rate. It's actually written into law. Hospitals will actually get reimbursed at a higher rate if they meet certain performance metrics, and infection control is actually one of those performance metrics. And if they can get if they get a higher reimbursement rate from Medicare, a rising tide, as they say, floats all boats. And so if you get a higher rate from Medicare, even though you have to invest in it, it will be extremely competitive from the hospital's perspective to lower that infection rate because they will get a higher reimbursement rate. That law is set to take effect in October 2012. So we have approximately 18 months until we see whether or not hospitals are actually going to go aggressively towards controlling hospital-acquired infections, and they are being incentivized. The director of the CDC has actually declared war on healthcare-acquired infections. It's one of his top 6 things that he needs to go after, behind HIV, automobile accidents, as well as teen pregnancy. So I think the country is serious about controlling healthcare infections, and any methodology that will reduce infections, I think hospitals and the healthcare community will beat a path to their door.
You know, what I find very interesting from a philosophical perspective, looking at, you know, the whole notion of antimicrobial control, antibiotics, and the like, is that here it is, a metal that the Phoenicians understood was antibacterial, and we've literally spent hundreds of billions, if not trillions of dollars into new antibiotic drug discovery to only come up with the notion that it's not in the best interest of pharmaceutical companies from an economical standpoint to continue to invest in those dollars in that area of research, when in fact there are ancient solutions that if people, microbiologists, would look at it, well, not microbiologists, but people that understand this stuff, could really bring something as inexpensive when you look at the cost of copper relative to $1 billion to bring a new drug to market, to actually effectuate change. Because the bottom line is that we're running out of antibiotics to treat patients with bacterial infections. So, you know, I more power to your research, Michael, because it seems to me that this is actually a very interesting and intriguing possibility as a way to really effectively, if it works, to reduce the incidence and actually possibly kill a lot of the antibiotic resistant or multiple antibiotic resistant strains that are wreaking havoc throughout the U.S. in hospitals and nursing homes.
The microbial world is very much like real estate. It's all about location, location, location. And where these microbes are in the hospital environment really drives home a lot of these issues. When you consider that how much disposable material is actually used in a hospital today, most of that is due to fighting transference of microbes, because it's so challenging to eliminate microbes, and you know that's why we have disposable everything. Microbes are the original Doritos factory. You know that old ad, "We'll just make more." Well, that's what effectively bacteria do. They make more, and we really have to be clever and listen to what the bacteria are telling us. They're telling us, you know, we gave you 100 years of oil, we gave you 100 years of antibiotics, and antibiotics came into substantial use in 1950, and if you do the math, we're going to about 2050, and if you do the law of returns on how many antibiotics we're developing each year, you can see it's pretty clear that come 2050, unless we really get serious about redoubling our efforts at developing antibiotics, we're going to be struggling. So we're going to have to go back to what the ancient Phoenicians did in terms of controlling infections. One of the things I tell my medical students and dental students, I ask them the rhetorical question, why is your mother so terrified when the physician tells you you have a strep throat? And the answer is, at the turn of the 19th to the 20th century, 8 out of 10 people who developed a strep throat died. Today, no one has memory of that death rate of 80% or higher. Today, because of penicillin and the effective antimicrobial therapy that was developed in the 20th century, strep throat as a killer is effectively forgotten. And so I think it's part of our, if you will, grandmother's genetic memory that we're so fearful of strep throat, even in the age of antimicrobials. But as we all know, bacteria are constantly adapting, and we're seeing it in the clinic each and every day. So I think we have to go back and rediscover the literature that we may have forgotten in order to redouble our efforts at controlling infections and actually coexisting with our microbial friends, as well as our microbial enemies.
So I understand that copper does not work as an antimicrobial if it's wet. So your water pipes are not sterilized by the presence of copper. Do we know why that is?
I think it's contact, because you have to, if you go back to thinking about the mechanism, if you're suspended in solution, remember, consider how small bacteria are and the water flowing through the pipe. But there is data out there. There's an individual in Virginia whose name just left my head, who's done a lot of studies on water pipes, asking the question, what's better, a copper pipe or a plastic pipe to transport potable water? And he has data out there showing that transporting water via copper is much better because you don't have to use as much chlorine at the injection point where the water leaves the water plant. And you have to use more chlorine. So there actually may be some residual antimicrobial activity for the microbes as they tumble through the pipe. Because it really makes no sense, because once a bacterium sees a metal, it'll go to that infinite ground. At least it should, thinking thermodynamically.
All right. I think that's a good discussion of that paper. Now, what I plan to do on TWiM at this point, after we've talked about papers, is to read email from our listeners. And I encourage everyone to send in your questions and comments. We do that on TWiV and TWIP. It works beautifully and allows us to engage everyone. Of course, since this is episode 1, we don't have any questions from listeners yet. I'll tell you how to do that in a moment. But I brought in one email that I received on TWiV, and this is really an email for TWiM. So I thought I would read it and see if either Cliff or Michael had a suggestion. This is from David, who is a microbiologist in training at North Carolina State, not too far from Michael. And he says, would you have any recommendations on a good primer into microbiology that I can read or listen to? I am interested in extremophilic organisms. Thank you. So do either of you have anything to recommend to him?
I am personally biased in that I really enjoy Elio Schechter's book, The Microbes. And it's a really approachable text in which you can actually sit down and read it. It's very provocative in terms of questions that it asks. But the other thing I would suggest to the listener is to consider looking at Elio's blog, Small Things Considered, because Elio and his fellow bloggers cover a wide range of topics in microbiology in a very approachable manner in which he really distills it down to an essence that you really get a sense of what's going on. And then if you're interested in that area, you can dig deeper. And being a fan of social media and the internet and Google, you can, of course, Google anything that is on Elio's blog, and you'll learn an awful lot, because his topics are really outstanding, and he really covers the waterfront of microbiology on that blog.
All right, very good. Thank you, Michael. Good suggestion. There you go, David. And in fact, we hope to have Elio on TWiM at some point in the future, and that would be just great. Well, that'll do it for our first episode of TWiM. At the moment, you can listen to TWiM at microbeworld.org/TWiM. We are applying to the iTunes store and the Zune Marketplace for putting TWiM episodes there, and as soon as that's available, we'll send you the link as well. There is also an app which you can use to listen to TWiM on your iPhone or iPad or your Android device. Go over to microbeworld.org. There's a link to that app, so you can stream the episodes while you're carrying your phone. You can always, as I said, play or download the episodes at microbeworld.org/TWiM. If you like TWiM, tell others about it. Links and reviews in iTunes are particularly helpful. Send us your questions and comments. Tell us what you want to hear. I'll tell you how to do that now. You can send an email to twim@twiv.tv. That's twim@twiv.tv. Or you can go to microbeworld.org/TWiM, and there's a place where you can send an email there. And finally, if you have a story you would like to submit for us to discuss on TWiM, add it to microbeworld.org and tag it with TWiM. If you go over to microbeworld.org, you'll see that you can add stories and add tags to them. And if you add the tag TWIM, then we can find it and think about using it for a story discussion. I'd like to thank everybody for participating today. Stan Maloy, of course, who is at San Diego State University. Cliff Mintz, thank you for joining us.
It was a pleasure.
Would you like us to tell folks that you are at biojoblog.com?
That's correct.
There you go. Check out biojoblog.com, where you can read what Cliff thinks about the bio industry. Michael Schmidt, thank you for joining us.
Thank you, Vince. It's been a lot of fun.
Michael is at the Medical University of South Carolina, and we hope to have both you and Cliff on many future episodes. I am Vincent Racaniello, and you can find me at virology.ws. I'd also like to thank the American Society for Microbiology for supporting the launch of TWiM, Communications Director Barbara Hyde, and especially Chris Kondayan and Ray Ortega for their organizational and technical help. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.